I'm Annika, and I'm going to be talking about concurrent neural induction and anterior-posterior axis development. During neural induction, the mesoderm induces the ectoderm to form neural tissue, and then the neural tube is formed, resulting in the neurola stage of the embryo. In an experiment by Mangold, he transplanted different sections of the neurola into a younger embryo, and this resulted in a second occurrence of various structures. When the most anterior portion of the neurola was transplanted, this resulted in a second forebrain, while when the most posterior part of the neurola was transplanted, this resulted in a second tail. This showed that consecutive segments of the neurola induced successive anterior/posterior segments of the embryo. There are two possible models that could explain this phenomenon: the sequential model and the concurrent model. In the sequential model, the mesoderm first induces the neural tissue, and the neural cells decide whether they will induce more anterior or posterior sections later. In the concurrent model, the mesoderm induces neural tissue, and the neural cells already know whether they will be inducing anterior or posterior sections. So, which model is the correct one? From his work, Mangold proposed that the correct model was the concurrent model. However, it turns out that the answer isn't so obvious, and there have been a lot of conflicting opinions. Nyaku proposed the sequential model, also known as the activation transformation model. All cells start out by default with an anterior axial identity, and later on the cells are transformed into more posterior identities. This has been the accepted model for decades, and was supported in various studies. One such experiment involved manipulating the amounts of Wnt, FGF, and RA in a culture of neural plate tissue. They found that manipulating these could actually alter the resulting axial identity of the tissue, supporting this hypothesis. However, these experiments were mostly done to determine axial identity within the brain, and were extrapolated to also apply to more posterior portions like the spinal cord. In fact, recent work has begun to disprove this model, favoring something closer to the concurrent model instead. Recently, it has been discovered that the spinal cord cells are actually induced by cells called neuromesodermal progenitors. These cells reside in the posterior epiblast and are continually producing spinal cord cells and somites after gastrulation. When embryonic stem cells were cultured with Wnt, they formed neuromesodermal progenitors that could generate the spinal cord. But when they were cultured without Wnt, they formed neural progenitors that were unable to generate any cells past the most anterior portion of the spinal cord. This disfavors the activation transformation hypothesis, since these progenitor cells seem to be both necessary and sufficient for spinal cord induction, and also suggests that Wnt may be present in the posterior epiblast. However, there was still the possibility that these cells might go through an anterior intermediate state before forming spinal cord cells. This hypothesis was disproved definitively by an experiment by Metzys. In this experiment, she showed that cells first adopt an axial position and then acquire a neural face specific to their position. So, the activation transformation model only applies to the brain and not to the spinal cord. This means the anterior or posterior fates are already set by the time gastrulation has finished. Crucially, none of this evidence disproves the activation transformation model when applied to just the brain. So, what is this new model of induction, and how does it compare to the old model? In the anterior portion of the epiblast, Wnt inhibitors and BMP antagonists work together to create the anterior neural plate. However, different from the old model, there is also Wnt and FGF present in the posterior epiblast that generates neuromesodermal progenitor cells. In both models, the anterior neural plate goes through posteriorizing signals that subdivide the plate into forebrain, midbrain, and hindbrain. Notably, the new model does not include a transformation of the anterior neural plate into the spinal cord, as the spinal cord is formed from the neuromesodermal progenitors. Overall, this work supports the concurrent model of neural induction and AP development, that the mesoderm is differentiated early on between anterior and posterior, with the neuromesodermal progenitor cells resulting in posterior identity. Thanks for watching.